Welcome, it's Nancy from the County Library, the South Jordan branch, and I'm here again today with my husband, Dr. Philip Moos, who's a professor at the University of Utah in the College of Pharmacy. And we're going to dig a little deeper into DNA that he talked about last time.
Hello. So when we isolated DNA from strawberries, I told you that DNA was an instruction manual. But what I didn't tell you is how those instructions actually become the strawberry, or how those instructions encode for a human. So today we're going to go a little bit deeper into that. So DNA is basically divided into genes, and those genes then are transcribed into RNA, and that RNA is then translated into proteins, which are the machines and structural elements that actually make everything.
So wait a minute, let's back up a minute. So when we pulled the DNA out of strawberries last time, it came out in a big clumpy thing, if you remember. So what does DNA really look like?
So you may have heard of DNA as being a double helix, and what I've tried to sketch out here is basically that sugar phosphate backbone of DNA. So you have the two strands, and in between them then are the bases, and the bases are basically the rungs of the ladder. So if we took that double strand of that double helix of DNA and we straightened it out, it might look something like this. So we printed off these manipulatives. They were constructed by Eagle Knot, and they're available on Thingiverse, but it's a nice way for us to be able to look at what DNA might look like up close. So what you can see here is the sugar phosphate background. So the P's are the phosphate, the D's are the deoxyribose, the sugar part of it. You can see that one strand is going in one direction, another strand is going in the other direction. And most importantly, though, we can see how the bases are pairing. So A's always pair with T's, and G's always pair with C's. And so this is what makes so that we have a consistent code within the DNA to encode the genes.
Okay, so this is what DNA looks like, but what about the transcription that gets to RNA?
Okay, perfect. So the way that this works is that we need another molecule to come into place. So if we remove one strand of this DNA, and if we can substitute that with an RNA strand. So RNA is very similar to DNA. It has a different sugar, so this is an R now, the ribose, but it also has that sugar phosphate backbone. And now, instead of having T's, wherever there was a T, there's a U in RNA. But otherwise, they use the same bases. So from a strand of DNA, you can encode that strand of RNA to make a new piece of RNA, and that's how the gene in DNA gets transcribed to RNA.
Okay, so now we have an RNA, but to get to a protein, you said that we have to do translation.
Right. So let's move the DNA out of the way. So the way that this works is that another RNA molecule is used to actually do this translation event. So if we have some of these, we'll call these amino acids, and they're forming the new protein, and so we have tRNAs that then match sets of 3 bases, so that makes a codon, and if that codon encodes a specific amino acid, then that amino acid then gets added on to that protein. Add another amino acid. And so you just build up the amino acids, and then those can fold into the proteins. The proteins are the engines and structural elements that make up the cell.
Thank you, Dr. Moos, for that informative demonstration on how we get from DNA to a protein.
You're welcome.
I'm Nancy from the County Library, and you can reach us at thecountylibrary.org.